enthusiasm I'm looking for. It's the weekend. You have to study a little bit, maybe, for the exam on Monday. But it's still a weekend, so it's okay. We're going to finish up today. We're going to start photosynthesis. We're only going to get halfway through photosynthesis. So you're only going to need to know the light reactions for the exam. So it's a little bit less you have to worry about.
Maybe we should go over our notes.
All right, let's settle down. Normal set of announcements.
I don't have any.
Your final midterm is on Monday. It's going to be through glycolysis. There's a 4-cycle fermentation, citric acid cycle, electron transport chain. Don't forget about pyruvate oxidation. That's in there as well. And through the light reactions of photosynthesis. So whatever we get through today, plus a little bit on genetics and membrane transport proteins. So that's what's on the next exam. First thing Monday morning, make sure you get a lot of sleep, have a good breakfast, get your coffee in, you'll be ready to go. All right, so if you had recitation yesterday, you've had a review session already. If you have it today, that's your chance to get your review session. I encourage you to all study together, quiz each other to help each other prepare for the exam over the weekend. All right, since it is over a weekend and it's first thing Monday, if you have any questions over the material, try to catch me today. Okay? Any questions before we get started? About what's on the exam? What to expect? All right. This is what we just covered. So we finished on Wednesday. Glycolysis, glucose to pyruvate, pyruvate oxidation. We looked at what happens if there's no oxygen, fermentation pathways. With oxygen, then we looked at what happens in the mitochondria, that pyruvate oxidation, citric acid cycle, electron transport chain. All right, make sure you know those. Glycolysis, know all the steps, all the structures for all of the components, each of those 4 pathways. Know what you start with, what you end with, and what you produce along the way. All right, so just to get everyone's brain started. Hope the clicker is working today. Let's start with a review, glycolysis and cellular respiration. First question. When glucose is metabolized by aerobic respiration, what happens to the carbons of that sugar molecule? Very subtle. All right, everyone get their answers in? Let's take a look.
Right? That's all I got.
Oh. Take a look at what this last thing. Everybody wanted the answer that we shouted out. All right. Why is it oxidized? Loss of electrons. We're oxidizing it and giving those electrons to the electron carriers that we're sending through the electron transport chain. All right? gave you that answer? This is a freebie. Where do those electrons go? All right, everyone get that answer in. Let's take a look. All right, a little bit of confusion. What are our two electron acceptors in cellular respiration and glycolysis? All right, so the correct answer is D, because it's NAD. We're giving our electrons to NAD plus and FAD. One more. What's the final electron acceptor of cellular respiration?
I might have to do that because like my skin got but I don't know. Really? Why? Right? Right? Or is it
What's picking up those electrons at the end? All right, get your answers in. Let's take a look. All right. Class has it correct. Oxygen. All right, aerobic respiration. Oxygen is our final electron acceptor. All right, so that's a quick review of what we covered on Wednesday. Let's move now into photosynthesis. Just like with glycolysis and cellular respiration, for photosynthesis, you want to keep track of what we're starting with, what we're ending with, and where are the electrons moving. All right, it should be feel very familiar now that we've covered the electron transport chain. Because the whole goal of this, we're moving electrons and protons around to set up gradients to do work. All right, so photosynthesis, what are we starting with? What are our substrates? Carbon dioxide. Water. What's the third thing that we need? Light. All right, so in photosynthesis, we're using light for energy. We're starting with carbon dioxide and water. It's going to seem familiar to electron transport chain, only in the other direction. Electron transport chain, we're breaking down sugars. Photosynthesis, we're making sugars. What are our products? Sugar and oxygen. All right, so what we're going to talk about are how we get from where we start to where we end, and what happens along the way. If you look at our plant cell, how many people have looked at a plant cell under a microscope before? A few. They're mostly composed of giant vacuoles, right? Everything's squished to the side. The particular organelle where we're going to be concerned for photosynthesis is the chloroplast. Those organelles that have the chlorophyll that make the plants look green. So first, let's look a little bit at chloroplast structure, the structure of that organelle. Remind ourselves what it looks like. Just like the mitochondria, has two membranes, an outer membrane and an inner membrane. We call the space inside that inner membrane the stroma. In mitochondrion, it's called the matrix. Chloroplast is called the stroma. However, in chloroplast, remember, we have an additional membrane-bound structure inside the organelle. We have those thylakoids, those membrane stacks. It's the thylakoids where the chlorophyll are. So there's an additional membrane to be concerned about, which means there's an additional space. So now we have the space between the outer and inner membrane. We have the stroma between the inner membrane and the thylakoids, and then we have the thylakoid space. You have to keep track of where things are happening in each reaction. Right, just for comparison, the mitochondria. Move this out of the way. Maybe. All right, outer membrane, inner membrane, the space between the two membranes, the matrix stroma, and we have that additional thylakoid membrane and space in the chloroplast. So keep track of where things are happening. All right, so look at photosynthesis. It's happening in the chloroplast. Photosynthesis is a set of linked reactions. It's going to be a little bit different than in cellular respiration, where we sort of a linear pathway through. There are the first set of reactions are the light dependent. They require sunlight. So these are happening in the thylakoid. We require light, water, and oxygen comes after the product. And the second set of reactions, the dark reactions, we start with carbon dioxide and we produce carbohydrates. All right, so we have the light reactions, and then the dark reactions are the Calvin cycle. So another cycle that we're going to learn, but that'll be after the next exam. The light-dependent reactions, that's what we start with, and what it produces. It also produces ATP and NADPH. So that energy currency of the cell as well as an electron carrier. Because there's an electron carrier involved, it means we have to regenerate, just like we did for glycolysis. We need a steady supply then of NADP to keep that reaction going. What was our electron carrier in glycolysis? NAD. So what does that P stand for? Phosphate. So there's a slight difference in electron carriers between cellular respiration and photosynthesis. So NAD was nicotinamide adenine dinucleotide. And photosynthesis, we have nicotinamide adenine dinucleotide phosphate. Right? So pay attention to whether there's a P on that electron carrier or not when you're reading your questions on your exam. All right? No, you do not need to draw NAD or NADP. You just need to know which is used in which reaction. Okay? All right, so those are our light-dependent reactions. Sunlight, water, NADP, ADP, and inorganic phosphate. And we get oxygen and ATP and NADPH out the other side. The linked set of reactions, the Calvin cycle, they don't require light. They're using CO2. They're using that energy produced in the light reactions to produce starch and sugar. And regenerating our electron carrier for us. So there's our two set of linked reactions, linked through those energy molecules and electron carriers. Notice that the light reactions happen in the thylakoids, the Calvin cycle, and the stroma. We're only going to get through the light reactions today. So as far as the Calvin cycle, all you need to know is what it starts with, what it ends with, where it happens, and how it's linked to the light reaction. You don't need to know the specifics of those steps involved. Okay? For the light reactions, let's take a look at how these happen. Here is a set of questions to help you parse out the important information. Just like we did in an electron transport chain, what are we starting with? What are we ending with? Be able to explain generally how it works. We're going to talk about photoreduction and photophosphorylation. Understand what those are. And I think today we'll probably only get through non-cyclic photophosphorylation. We'll see how far we get. If we don't get too cyclic, don't worry about it. Okay? Pretty straightforward. Follow the electrons. Okay, so there's our scheme. This is all happening in the thylakoid. What part of the thylakoid do you think we're working at? The membranes. All right, this is where it's going to start to feel very familiar to the electron transport chain. We have protein complexes embedded in a membrane. In this case, it's the thylakoid membrane, so things are happening between the stroma and the thylakoid space. We have linked sets of protein complexes, starting with photosystem 2. I know that's confusing. But you start with photosystem 2. So the players are photosystem 2, plastoquinone, a cytochrome complex, plastocyanin, photosystem 1, ferredoxin, and ferredoxin NADP reductase. So those are the major components, and we're going to look at what happens in each one of those, what their jobs are, and how the electrons and protons are moving through these complexes and across the membrane. Just like in the electron transport chain for cellular respiration, the system is set up to move electrons in order to achieve making either electron carrier or ATP. All right, so you're going to see this again. First, let's talk about the photosystem. All right, this bit is different from the electron transport chain. So let's look at what the photons are required for, where the electrons come from in this whole system, and where the electrons end up to start off with. So first of all, where do the electrons come from? What are one of our substrates for the light reactions? Light and water. So we're stripping electrons off of water. Question? No. Just the electrons. The protons, we'll see, end up So we're breaking apart water into oxygen, our final product, electrons, which get sent through the photosynthesis, the light reaction, and then protons. And we're going to try to set up a proton gradient just like we did in electron transport chain. Okay, so we'll look at that in a minute. So if the electrons are coming from water, where do they end up? In our electron carrier. They're going the opposite direction of cellular respiration. Cellular respiration, we're taking electrons from our electron carriers, and they're ending up in water as a final electron acceptor. In the light reactions of photosynthesis, we're taking those electrons off of water and putting them into electron carriers. All right, and we require this whole process. I have to show you yet to do that. So when in the electron transport chain, when those electrons end up being picked up by oxygen, what's their energy state? High or low? When they're picked up with oxygen by oxygen at the end of the electron transport chain? Low. We put them in a high energy state, and as they're passed from protein to protein, they lose energy along the way. We're stripping them off of water now. So what do you think their energy state is? Low. So what do we need to do to these electrons? Give them some energy. All right, that's where those photons come in. Those photons provide the energy that excites those electrons into a higher energy state, and then they can be passed down from protein to protein, similar to the electron transport chain. All right, so we see in photosystem 2 where we start, we get those electrons. Our photosystem picks up the energy from photons of light, excites the electron. That gets passed between proteins. That cytochrome B complex pumps protons into the thylakoid lumen, the thylakoid space. Those electrons get transported to photosystem 1 once they've lost energy. In photosystem 1, pick up more energy from another photon of light to excite those electrons again. Then they're passed through a series of proteins and finally picked up by NADP. So the photosystems are designed to catch photons of light and pass that energy to electrons to bump up their energy state and then strip that energy back off by passing them through a protein chain, transport chain. So let's look specifically a little bit at what's happening in these photosystem, the centers where photons of light are being absorbed and transferred to those electrons. In the light-dependent reactions, the pigments that absorb energy of light are chlorophyll. That should sound familiar, I hope. Chlorophylls, there are two types of chlorophylls, generally A and B. They both have a similar structure. There's this ring structure that absorbs light and sticks out of the membrane, and then this tail structure that anchors that into the membrane. If we look at the wavelengths of light chlorophyll is absorbing, so here on the y-axis is amount of light absorbed, x-axis is the wavelength of light with the color to help you translate that. These are absorbance curves for each pigment. Additionally, these photosystems have accessory pigments, carotenoids. Also notice they have ring structures that absorb light connected by a long tail. The chlorophylls absorb blue light and red light, which means they transmit green. Why plants are green? Thank you, chlorophyll. So they're using blue and red light picking up those wavelengths of light to power photosynthesis. Carotenoids are picking up blue and green, so carotenoids are absorbing blue and green, so they are generally, something filled with carotenoid looks yellow or orange. Okay? These chlorophylls and the carotenoids are organized together into clusters that we're calling these photosystems. And there are two major components of a photosystem. There's an antennal complex, so all these pigments are on the outside, and then there's a reaction center. We look at the job of the antennal complex versus the reaction center. The antennal complex, like it sounds, it's an antenna. It's picking up wavelengths of light. So all of these pigments surrounding that reaction center, they're absorbing photons of light, and then they're passing that energy from one pigment to another. They're not passing electrons, they're just passing the energy. When that energy gets to the reaction center, that's when we get the electron excitation and the electrons being passed, then starting down through that protein cascade. So the antenna complex, the job is just to absorb energy and pass it through a process called resonance from one pigment molecule to another and get it to the reaction center where it can be used to excite electrons. Once it gets to that reaction center, that's a specialized chlorophyll molecule that acts as an electron acceptor and picks up those electrons to start the transport chain. All right, so not until it gets to the reaction center, those electrons start moving between the proteins to accomplish work. And that happens in both photosystem 1 and photosystem 2. Photosystem 2 and then photosystem 1. So if we look at how electrons then, once they're excited in a photosystem, and these photosystems are linked, as I showed you in a few slides ago, electrons come from water, move through photosystem 2, through plastoquinone, through cytochrome, through plastocyanin, to photosystem 1, where they pick up more energy, through ferredoxin, through the reductase, to NADP, to form NADPH. All right, so let's translate that into a more simplified diagram. So here's the diagram we started with, the major players in the light reactions. Remember, stroma, membrane, thylakoid space. So can you draw this figure? And draw arrows to show how the electrons move between these different proteins and protein complexes. And not only are the electrons, we haven't talked about yet the protons. So where are the protons moving? Which protein is moving the protons and in what direction? How do the electrons move through these protein complexes? I'm going to give you a few seconds to think about that, and then we'll take a look at it. Okay. Where do the electrons start? PS2. Where do they end up? PS1 to the NADP reductase. All right, what we haven't talked about yet is that it's the cytochrome B that's moving protons. All right, so it's using those electrons as it passes them and the energy they have to move protons from the stroma through to the thylakoid space. So in red is the movement of electrons all the way through to NADPH. In pink here, the movement of protons. So if we're moving protons, what are we doing? We're creating a gradient. Starting to sound familiar? All right. If we look at where these electrons are coming from, remember we're stripping them off of water, so this is Bailey's question earlier. Whether we're counting those electrons with hydrogen protons or not, we're just sending those electrons through this pathway. The protons, so we end up with oxygen, and then a bunch of protons in the thylakoid space where the water is being split. So not only through this process is cytochrome moving protons across the membrane, we're also creating them in that thylakoid space when we hydrolyze water and take those electrons. So we're stacking up that gradient with high protons on the inside of the thylakoids, fewer protons in the stroma. So what does that mean if we want to make ATP using this gradient we just set up? What kind of protein complex do we need? What's it called? ATP synthase. Which way the proton's going to move to make that ATP? Out of the thylakoid, so across the membrane from the thylakoid space into the stroma. Where is the ATP going to be made? What side? In the stroma. All right, so the final step in this process, we have that ATP synthase, protein complex. Protons are going to move through that. And that complex will use that energy to make ATP. All right, that is essentially the light reaction. Splitting water, taking the electrons, using photons of light to excite those electrons to a high energy state, passing those high energy electrons from protein to protein, stripping off a little bit of energy along the way, moving protons across the membranes. They get passed to photosystem 1. They get excited again with another photon of light. And then at that point, they're passed through a series of proteins and finally end up in our electron acceptor. Question, David?
Yeah.
How many protons are created by H2O molecules? Oh, we're not counting protons. We're not counting sort of a direct. All right, so in the electron transport chain for cellular respiration, we translated our electron carriers into how many ATP they're producing, right? We're not making that same kind of calculation here. All you need to know is that we are making a lot of protons, both by bringing them across the membrane and by splitting water. And then we're making ATP as those move back across the membrane. Yeah?
So is it still 3 to 1 though? 3 hydrogen to 1?
That's a good question. I'm actually not entirely clear if the protein synthase that works in photosynthesis has that same sort of ratio? I would assume it's similar, but I don't know for sure. It's a good question. You don't have to keep track of it quite the same way. All right, so the processes that we're talking about here, unless anyone else knows, anyone know how many protons it takes to move across the thylakoid membrane to make an ATP? Who's got their biology trivia down? No one? All right. I don't know. I'll see if I can figure it out. So the processes we're talking about here. So the first one, photoreduction. Photo, we're using light energy. What molecule are we reducing? What's gaining the electrons? NADP, our electron carrier. So our photoreduction process is the light-dependent synthesis of NADPH. The other process, photophosphorylation. What is it that we're phosphorylating? ADP to ATP. Photophosphorylation, the light-dependent synthesis of ATP. All right. Those are the light reactions. Be able to follow the electrons, know what you start with, what you end with, where the protons are moving, where they're coming from. Are there any questions? You should be able to draw that, yes.
What?
You should know the specific names of the complexes.
PXY.
There's only 7 of them. 2 of them are the same, photosystem 2 and photosystem 1. Are there any other questions over this or any other process in glycolysis of cellular respiration? Now's your chance. So Bailey's question was whether or not you need to track the numbers of things like we did in cellular respiration. I don't expect you to know sort of how many protons are moving back and forth, how many ATP are produced, etc. It's harder to track in this system because it's not a direct input to output. As you'll see, there are several pathways. We'll talk about after the exam another pathway through these light reactions. So it's not always a straightforward water in, ATP out. So you don't need to keep track of number of protons, number of ATPs. Okay, does that answer what you were asking? Yeah. You need to know how they're moving and what's produced, but I don't expect you to know the exact numbers. Yeah. Everyone settle down, we're still asking questions. So the question is the electrons that start the electron transport chain in cellular respiration. So where do the electrons that start the transport chain for cellular respiration, where do they come from? What are electron carriers? And NAD, AD. And where do they start the process? What component of the mitochondria?
Cytosol?
It's in the mitochondria. What membrane is it moving across? The inner membrane. Which way are the protons moving? Out of what? Everyone's doing this.
Out of no, moving into the matrix, out of the
So where do they have to start then?
matrix. Starting in the matrix, it comes out.
Did you hear that? So starting in the matrix, pumping things across it, set up that gradient, and then they're flowing back. Yeah? Well, so for both, essentially. Yeah? I believe that's right. Any other questions? All right. We'll finish a few minutes early so you can all rush home and practice. That way. If you have any questions, let me know.